Hello, everyone. Welcome to the second part of more the innate system. So, in the first part, we spoke a lot about, the physical barriers, the chemical barriers, let's say the first line of defense. That is still part of the innate immune system because they're non-specific. They act like, literally on the spot within minutes. And they're there basically to kind of help us against a variety of different organisms or microorganisms, sorry. So, it's non-specific. So now let's move on to more the cellular, the more, let's say, the cell kind of the innate immune cells and how they can actually protect us. Because let's imagine now that the microorganism, be it the bacteria or the virus or the fungi or whatever you're basically infected with, penetrates. It actually managed to destroy the first line of defense. Now it's in your blood or it's in your skin, or let's say past the skin. Okay? And then now comes literally the time of your innate immune cells. So in today's topic, we're going to discuss four things, mostly about PAMPS, pathogen recognition, and PRRs, macrophages in the tissues, phagocytosis, and inflammation. So as I said, first thing that has to happen is that the bacteria has to, of course, get inside, right? Or whatever microorganism gets in. Once it gets into there, the bacteria is literally, as you can see, this smiley little guy wants to go inside to the cell and literally kill it. We need our innate immune system, our innate immune cells have to be able to recognize this bacteria. It's extremely important for them to do so. But how is a phagocyte, or an innate immune cell, able to distinguish between this bacteria and this healthy cell? Because phagocytes or innate immune cells don't have a brain. They need to be able to recognize it though still. So there has to be some sort of mechanism in where innate immune cells have to really come in and take over. Because if they don't, the bacteria is going to be able to actually multiply and actually harm us. Because if you think about it, a bacteria in the perfect conditions, like in our blood, can multiply every 20 minutes. So imagine you're infected by two bacteria, then it becomes literally four, then eight, then goes on and on. And in a couple of hours, there are hundreds of thousands of them. So the innate immune system has to be able to recognize them extremely fast. It has to really be quick in doing so. So there has to be some sort of pathogen recognition. Well, basically, the most important thing, as I said, is that your immune cells have to be able to detect kind of pathogens. And they have to detect it in a, let's say, non-specific manner because we are infected by a huge variety of bacteria, a huge variety of viruses, of fungus, of parasites. They all have to be able to actually able to kind of, let's say, be able to kind of recognize all of them. But how is that possible? Well, through evolution, certain, let's say, bacteria, viruses have evolved in a similar manner, despite them being different, let's say, species or whatever. They actually have certain, let's say, molecular patterns or motifs. And motifs, for those who don't know, or a refresher, motif is literally a repetitive structure. And you see this a lot, basically, within our own proteins, but you also see it with a lot of proteins or carbohydrates or lipids within a variety of microorganisms. And they keep having this really, let's say, repetition of them. So what the innate immune cells actually have are host receptors that are able to recognize these patterns, these molecular patterns that are literally associated with microbes. And we're going to talk a lot about, different types of patterns and different types of literally host receptors. So literally a pathogen comes in and through certain, let's say, molecular patterns that it has, the innate immune cell is able to detect it and actually be able to kind of recognize it that, hey, this is something foreign. I need to kill it. So, what are these literally, molecular patterns? Well, we call them PAMPs. They're called pathogen associated molecular patterns. The name is so giving. It literally tells you what it is. They're literally pathogenic, literally they're pathogen associated, so they're associated to a pathogen, molecular patterns. So they're literally a molecular pattern that you see. This could be, for example, certain carbohydrates. This could be lipoproteins, lipopolysaccharides. These things that are literally components of bacteria, fungal cell walls. So those are what it can be, but it can also be even to the different type of nucleic acids during bioreplication of bacterial DNA. Because viruses and bacteria, even though they have literally very similar nucleic acids as we do, they have different patterns. This could be different methylation status. This could be different structures. For example, some viruses have double RNA strands, which we don't have, for example. These different things that to an immune cell, or specifically an innate immune cell, is foreign and is able to distinguish it from healthy and something that is, let's say, foreign material or pathogenic. So it's usually extremely, these are the PAMPs. And usually these PAMPs are actually, let's say, patterns, molecular patterns that are, let's say, quite essential for the function of a pathogen. So it's quite hard for those, let's say, pathogens to remove them. So, for example, in the case of viruses with double-stranded RNA, the virus will be like, oh, okay, damn, I'm being recognized because of my double-stranded RNA. But sure, but what are you gonna do? Are you gonna remove it? You can't. You really cannot do that. The virus is not gonna be then alive. It's not gonna be able to replicate. So thankfully, our innate immune system has been able to recognize very extremely important PAMPs that are really needed for the function of a pathogen to survive. So as I said, really these PAMs really help the host cells to distinguish between self and non-self. It's extremely important because if it's not able to do that, if the innate immune system is just there, just going crazy at whatever it's encountering, yeah, well, that's going to cause us a lot of auto-inflammatory and autoimmune diseases. Like you don't want that. You want it to be able to actually only be activated when we are in the presence of an invader or a pathogen, and it does it through PAMPs. Of course, there are other basically, let's say, molecular patterns. There can be non-pathogenic organisms. These are literally could be certain, let's say, bacteria that are actually not pathogenic. These could be basically other viruses that are not pathogenic, or these could be, for example, archaea that don't do nothing to us. And we call these basically microbe-associated molecular patterns. They're not pathogenic associated, they're microbe. Or we have DAMPs, damage-associated molecular patterns. These are literally the ones that I was saying. These are literally proteins or certain molecules from our own system that are in places that they shouldn't be. An example for this is ATP. ATP is always should be in the cytosol, but sometimes it is actually found basically in when cells are being killed, it's being released to the outside. If a macrophage or a true neutrophil or another innate immune system cell comes and actually detects it, it already can recognize like, oh damn, this ATP, why is this ATP outside? Something is wrong. So these are dams. These are things literally inside molecules from us, molecules from the basically from the host, or in our case from ours, that are in the incorrect places. Like DNA, DNA is also always supposed to be in the nucleus, should not be in the cytosol or outside in our basically blood. That is already also a damp. So these are called damage associated molecular patterns, things that usually when you damage a cell are released. And let's go to basically different types of PAMPs. For example, we can have bacterial peptides. This could be the peptidoglycan. There's basically very nice, let's say, cell layer that majority of bacteria have. Then you have the flagellin. Certain basically bacteria have this kind of flagella, which has it's literally made of flagellin. It's this protein that makes the flagella. It's this literally kind of tail for the bacteria that helps them actually kind of move. Certain carbohydrates, for example, mannose. If you guys remember with the complement system, the lectin pathway is activated through mannose. We do not have mannose. We do not have mannose in our cell walls, or sorry, not cell walls, in our cell membranes. So that already is a pathogen-associated molecular pattern that is found in not just in one type of bacteria, but in thousands of different types of bacteria. LPS, this lipopolysaccharide that is found mostly in gram-negative, basically bacteria. We don't have LPS, but as I said, you can also start seeing bacterial, basically DNA or viral RNA variants. For example, usually in our DNA, when we have basically these kind of the CpG DNA, which is basically just cytosine and guanine together, usually they're methylated when it comes to um, our cells. Methylated means it has a methyl group, CH3 group attached. But when it comes to bacterial DNA and viral RNA, no. And this is mostly because their own polymerases do not have the function to put a methylation. So if suddenly the innate immune cells actually recognize this unmethylated or kind of let's say sees some DNA and we sees that in the C and G areas there's no methylation, it's already a danger. You can also have basically double-stranded RNA that we know that shouldn't be there. In parasites, they have certain GPI anchors. In yeast, they have certain, what do you call it, structures like beta glucan. This is some sort of basically sugar that we don't have in our system and is very found extremely in yeast. I actually work a lot with beta glucan. I literally published a paper on how good beta glucan can be in actually activating our innate immune cells. It can be extremely potent, I would say. So now we spoke about the PAMPs, but what is the thing that actually, let's say, recognizes these PAMPs? These are the receptors. These are receptors that are found in our basically innate immune cells, but they can also be found in our normal cells. They can be found in any type of cell. Literally, it can be in your liver cells, it can be in your endothelial cells, they can be in your skin. Basically, they're there just to be able to recognize if something is wrong. It's not very specific just for the innate immune cells. They can be found also in different cells. But these are called pattern recognition receptors. I literally love biology because the names are so self-indicative. They're so easy to memorize, honestly. They're literally pattern recognition receptors. They're recognizing patterns, so which are the PAMPs, technically. So, as we said, these kind of sense for molecules like PAMPs. And usually they can be found either on the membrane of the cell or they can actually be found inside the cell, basically in the cytoplasm. And this is extremely important because depending on the pathogen, its location, because bacteria are majority of the time outside. So you want the PRR to be on the outside, so on the membrane. But viruses cannot live in the outside. They need to go inside. So you need literally PRRs inside the cell to actually really kind of, let's say, detect them. And once they detect, this causes a signaling pathway, which we will talk a bit about, in a way that it will activate certain cells, be it a macrophage or neutrophil or just a skin cell, to basically kind of be on alarm. In macrophages, it can actually tell them to start releasing a lot of cytokines and chemokines. So basically molecules that are literally alerting your innate immune system. I'll be giving you examples about cytokines and chemokines in a bit. But it can also tell the macrophages like, hey, start doing phagocytosis. So these PRRs, once they're activated, they're literally kind of inducing some sort of effector mechanism, be it phagocytosis or release of cytokines or chemokines. And as I said, these PRRs can also recognize DAMPs, these damage-associated molecular patterns that I was talking about, like ATP or DNA that is basically on the outside in the cytosol. So, as we said, once you have the PRRs are able to recognize and bind to the respective ligands. So, for example, let's imagine here you have these are different PRRs, which we're going to be talking about. You have, like, for example, this lectin receptor that binds to carbohydrates. You have this TLR that binds to different proteins, these scavenger receptors that also bind to different proteins on the bacteria. This actually then allows this, let's say, immune cell that expressing these PRRs to get activated, and many effects can happen. There can be a release of cytokines, chemokines, hormones, growth factors, different types of molecules that can actually have a anti-pathogenic effects. So things that are literally trying to kill your pathogen. They can actually also induce inflammation. It could really cause an inflammation, which we're going to be talking a lot about now, and how is that useful. It could also actually induce other immune cells, for example, phagocytes, to come in and actually eat up this bacteria, or actually bring NK cells to come in and release perforins to kill the bacteria, the ones that we were talking about in the first lecture of immunology. But it can also allow other cells, other phagocytes like macrophages and neutrophils to come and eat dead or mutated cells as well. So when it comes to PRRs, there's so many different families. There's so many different types, and we like to put them in different as families. And we put them as families mostly by their location or where they're expressed or the type of thing they're recognizing. And the most, let's say, famous PRR that there is, they're the toll-like receptors. They were the one of the earliest PRRs actually discovered. And actually, my PI, the person that I work with mostly, he and his during his postdoc actually in Houston. This is literally in, I think, if I'm not mistaken, Vincenzo was in Houston in the uh, late 90s, early 2000s. He was actually was there to try and discover different PRRs. So he quite knows them quite well actually. And he was telling he would actually teach us a lot about these toll-like receptors. And these toll-like receptors, all of them are literally these type 1 transmembrane glycoproteins, meaning that they're glycoproteins that are literally attached to a membrane only once. So they pass through the membrane only once, and they literally have basically their end terminus outside trying to bind. So they're all found in a membrane. But where in the membrane is basically a good question. It could be literally in the phospholipid membrane in the whole cell membrane, or they can be in the membrane of a vesicle. So they can be inside a cell, but it always has to be in a membrane. Either it's inside a vesicle, inside the cell, or on the outside of the cell membrane, basically. And of course, depending where it's on its location, it will be able to recognize certain, let's say, pathogens. If it's inside, it's most likely to be viruses. If it's outside, it's most likely to be bacteria. So these are extremely wide. There's like many different types. I think there's like what 10 different TLRs, and each one basically binds to different types of things. And they're, let's say, once they're actually, let's say, activated, they start to release a lot of cytokines. They express a lot of cytokines in general. But other than cytokines, they actually also will push macrophages to do actually phagocytosis, and it will actually do something called antigen presentation, which we will talk about next in the next weeks coming possible. But it literally helps them in phagocytosis once they're activated and the expression of inflammatory cytokines. Then you have a different type of a PRR family that's called the nucleotide oligomerization domain. There's node-like receptors. These guys, they really will bind to quite a very different types of pathogens. Either they bind to pathogens or their components, or as well as basically things that they're produced by the infection. These could be, for example, toxins or by injury. For example, they also bind to DAMPs. So this is what they mean by other components produced by infection and injury that are in the cytoplasm. And these are basically intracellular. These are intracellular PRRs. They're inside, and once activated, they express cytokines, extremely powerful cytokines for inflammation. So these are there's the 10 different types of TLRs that I was talking about. For example, you have this TLR2 that basically will only recognize glycolipids and peptidoglycans that are in gram-positive bacteria. Then you have TLR3, which engages in double-stranded RNA. Then you have TLR1 and TLR6, which basically binds to certain multiple dicycle peptides, which are only found in mycoplasma. 4 is in gram, it's more the LPS, which is in gram negative. 5 basically binds to flagellin. 7 and 8 to the single-stranded RNAs in viral replication. The TLR9 binds to bacterial DNA, and TLR remains an orphan receptor, meaning that it doesn't really know where it is actually recognizing, really. I'm not expecting you guys to actually memorize what every TLR does. What I'm expecting you to memorize is like what the toll-like receptors, where they're found, and basically what happens when they get activated. So, but this is just for your information. And of course, again, depending where they are, they have certain, they will bind to certain things. For example, the ones that are actually binding to bacteria, they're all outside. So TLR 1, 2, 4, 5, 2, and 6, that literally are all binding to bacteria, they need to be outside. The ones that are binding to DNA, they need to be inside because DNA is never going to be, you're not going to find DNA outside unless you have a damage. But when it comes to bacteria or viruses, and specific viruses, the bacteria is going to be, sorry, the DNA is going to be inside. So that's why you have the TLRs 3, 7, 8, and 9 that are mostly intracellular. They're the ones that are literally binding to different types of DNA. And as I said, as you can see here, there's type 1 glycoprotein that they're literally binding, going through the membrane once, as you can see here, and they always have to be in the membrane, either in the cell membrane or in the vesicle, but always. Then you have other basic PRRs. You have this retinoic acid inducible gene RIG1. These are extremely important for viral immune responses. These RIGs, actually, you can find them not just in innate immune cells, you can also find them in normal cells as well. They recognize viral nucleic acids extremely well. Then you have the C-type lectin receptors. These are only found in phagocytic cells, mostly like neutrophils or macrophages or dendritic cells. These are extremely good at binding to carbohydrates. They are really good at that, and they can induce literally phagocytosis. And then you have the absent and melanoma 2, so the AIM2-like receptors. These are also inside the intracellular, and they only basically recognize intracellular DNA. But these guys are very well known to release very specific cytokines like interleukin 1 beta and interleukin 18. I'll be talking a lot about interleukin 1 beta a lot actually upcoming because it's a very powerful cytokine for inflammation. But these are the different types of PRRs that exists that are able to recognize all different types of PAMPs. So as I said, you have membrane-bound receptors like the toll-like receptors. They always have to be on a membrane, or also the C-type lectin receptors. The cytosolic receptors are the ones that I spoke about, the node-like receptors and the RIG1 receptors. These are the ones that are literally binding to things inside, either the viral RNA or to certain peptidoglycans that are found inside. So again, sometimes by looking at where they are, you can actually sometimes really decide are they what they're going to bind to. If they're going to bind to DNA, then you're going to be like, okay, they are definitely inside. Of course, membrane bound or not, yeah, the TLRs that are inside can bind to DNA, but they're also in the membrane. So once they're activated, they basically are able to induce the release of cytokines and all these different type of things, or induce phagocytosis through signaling pathways. And there's three main types. There's the NF-kappa B signaling pathway. This is the most common type of signaling pathway. It's literally a series of proteins being activated, and these are extremely common. Then you have the mitogen activated protein kinase. These usually happens with PRRs that have CR9 or 309 in their membrane, or sorry, in their basically in their domains. And these are basically mostly are very good at releasing pro-inflammatory factors. So NF-kappa B and also this MAPK signaling are really good at releasing pro-inflammatory factors. Then you have this other type of signaling pathway, which is the TBK1 IRF3. And these are very important, mostly for viral innate immune responses. So this TBK IRF3, you mostly find it actually with RIG receptors. Because as we said, RIG receptors are very good at antiviral immune responses. So they really do kind of push this TBK1 IRF signaling pathway, which releases this type 1 interferons. These type 1 interferons are literally very good for antiviral infections. We will be talking about them later in much more details. Now, let's talk about phagocytosis, because as I said, phagocytosis is extremely, extremely important with for helping us to combat microorganisms. So, as I said, Phagocytosis is literally the name is phago is eating, cytosis is cell, so cell eating. It's technically eating up either uh, microorganisms, or these could be apoptotic or necrotic cells, cells that are basically have been damaged or already dead, to kind of remove them because you don't want them to be around, because if a dead cell is around too much, it can be releasing too many damps, which can really push our immune system a bit crazy. So what happens technically when it gets in contact with either a bacteria or a dead cell is that you actually have, let's say, this contact, and this contact with it is has to be through a PRR or through other basically types of receptors or the complement system or an antibody. The macrophage is not going to be able just to eat randomly. It doesn't do that. It has to become either by activation of the PRR or that complement the C3b or the C5b are literally all around the sorry, it's mostly the C3b, my bad. It's the C3b basically being opsonized on your bacteria or antibodies that allows then the macrophage to kill. It's not random, it is done through certain contact with the receptors. And this is primarily a eukaryotic process, meaning that prokaryotes don't do this. And what we've seen this a lot. If there's any literally defect in phagocytosis, be it in the receptors, be it in not producing antibodies or complement, or in generally that there's no lysozymes or there's no certain, let's say, mechanisms to do the phagocytosis, well, this can be extremely dangerous because it opens up us to many the risk of serious infections. So we're prone to get infected much more. And in generally, most of our literally current knowledge when it comes to phagocytosis is usually done through macrophages, actually. And that's just purely because macrophages are extremely easy to manipulate in the lab. Clinically, usually they use neutrophils because there's so many of them. But let's put it away, our knowledge towards the mechanism of phagocytosis is mostly through macrophages. When it comes to clinical studies, we use neutrophils in the case, but it's mostly, let's say, if you want to really study mechanisms, we go for macrophages. So basically phagocytes, there's many different types. So the most common types are monocytes, dendritic cells, macrophages, and neutrophils. These are literally your most ones. Monocytes are literally basically smaller versions of macrophages. Usually monocytes, they tend to basically to turn into macrophages a bit later on during their lifetime. Then you have dendritic cells. These ones are professional. They're called professional phagocytes. They are literally antigen presentation. They literally are the ones that are able to phagocytose bacteria, but they're also able to present it to your T cell to teach T cells what to kill. However, we'll be talking about them a lot next week. But what you have to know right now is that they're very good at phagocytosing. And then you have neutrophils. Neutrophils are there's so many of them. They're the biggest, literally, white blood cell population in your blood, and they are literally, how can I put it? There's so many. They're just a big abundant amount of them, and they basically have a lot of specialized TLRs or basically PRRs with them that can recognize multiple different types of parasites, bacteria, yeast, you name it. And they can basically these are quite cool because they can opsonize. They can literally internalize both opsonized and non-opsonized particles in some sort of way, and it's also the way that they are basically being able to eat phagocytes, their mechanism of how they're able to eat is not so well known like macrophages, because neutrophils, once you take them from the blood, they die within 6 to 8 hours. Macrophages, they can live up to literally weeks. So you're able to study them much more. Neutrophils, no, it's just a couple of days, honestly. But the whole mechanism of phagocytosis is quite simple. As I said, there's always has to be an identification. There always has to be binding towards a, let's say, a certain pathogen or that cell, and that's either through antibodies, through the complement, or through the PRRs. So once that basically is identified, this induces basically the membrane of the whole macrophage to internalize basically either the dead cell or the virus or bacteria. And then basically they internalizes it and it puts it in a vesicle that we call a phagosome. So the phagosome basically is the vesicle that's taken inside. And then you have literally a lysozyme. So these lysozymes are these small little vesicles that we have in our cell or inside the basically in the macrophage or the phagocytic cell that has all these digestive enzymes, specifically lysozyme. So what happens is that it fuses. Once it fuses, this induces actually the decrease in pH, and decreasing in pH causes the lysozymes to start to be activated. So once the lysozymes are activated because of the increase in acidic pH, you start to degrade, completely destroy everything inside. So this is the whole mechanism of phagocytosis.